Hi Nat5, so we're looking at photosynthesis and the stages that breaks down into. Unfortunately, it's a little bit more than you had before. Right, so in junior, first and second year, you had a basic overall photosynthesis. Okay, now the overall photosynthesis equation has not changed, but you've got quite a bit more detail in it. Okay, so what we have is a series of enzyme controlled reactions. Now that's important because as soon as I put that word in here, as soon as it says enzymes, you know there's going to be a temperature involvement here, because you know that enzymes have a link to temperature in terms of how well they work. But we're going to look at that later on. Okay, making glucose, and most importantly where it comes from, so we're going to use some raw materials, carbon dioxide and water. Now you can't build stuff up unless you've got an energy source to do it, and that's the light bit. So here's your basic equation: carbon dioxide and water and light, which is going to be captured by chlorophyll, makes glucose and oxygen. Okay, so that is your summary equation. There is nothing wrong with that equation. It's the same equation you've had for years, but this time you're going to have to be able to break it down a bit. Okay, so two stages. So before we look at that, got to look at where you actually have it. So where we have it is inside the chloroplast. Now, under the microscope, you can actually see individual chloroplasts. So each of these little green blobs inside here is a chloroplast. Each of these larger sections is a cell. So this would be in a photosynthetic leaf, probably from something like an LE3a, so a water plant, so you can get a nice clean look through. But each of these little green blobs is a chloroplast. This has got lots of chloroplasts in here, and generally we don't show all of them in a picture. You would have, you know, your diagram only has 4 or 5 maybe in a cell, but you can have them really densely packed. Okay, so the chloroplast itself is made up of 2 parts that you need to know, because there are 2 reactions. Okay, so if we zoom in on it, it's like a kind of circular blobby thing. We've got an outside membrane and an inside membrane. Don't worry about that. But what you have inside it are little discs inside the disc, as it were. And those little discs, each of these discs here, is packed full of chlorophyll. Okay, and you can see they kind of connect over to other discs. So it's a whole series of them. Okay, this is called the granum. And this is where we're going to do the light reactions. So this is where your colour is. The bit around the outside of this is not got any colour in it at all. It's just kind of goopy stuff, kind of like cytoplasm, but this is going to be specific for inside the chloroplast. And this is called the stroma. So it's going to have all the enzymes that you need to do the second part, which is called carbon fixation. Okay, so let's have a look at the first part. So the first stage uses energy from light. That's kind of obvious when it says light reactions. And the correct name for this would be photolysis, which literally means splitting. The lysis bit means break, and photo means light, so splitting with light. You also see it as the light dependent reactions, either way. Okay, right, so you're going to use light to smash up water. So absolutely clear, are we happy that water is really H2O? So I can draw that lots of different ways. Here's your kind of slightly more chemistry way, although that hydrogen is obviously dodgily large. So what we have here is something that we're going to use the light energy to smash this up. Okay, so it's going to break these bonds in here. And kind of obviously we're going to get oxygen and hydrogen out of that. We only need the hydrogen, we don't need the oxygen. Not for this reaction. Obviously we need oxygen to stay alive, but that's to use it in respiration. And you need to recognise there's a link between these 2 reactions. Okay, the plants which are making their food at the same time are producing oxygen, which they're not using for that. That's just an excess thing that they're releasing. But some of it, they'll just go, I'll just use that for respiration then. And the extra we get to use. Okay, what you need though is the hydrogen. And we also need some extra energy. Now the light energy is no good to us, but they can change the light energy into chemical energy, and you should know the form of that. That's ATP. Okay, so we're going to take water, we're going to take light energy, we're going to catch it with chlorophyll. Okay, chlorophyll, officially the term is it becomes excited. Okay, chlorophyll then batters water, smashes it apart, you get oxygen, which you then just diffuse out the cell, okay? And you get hydrogen, which, well, hydrogen on its own, if it's just a gas in pure hydrogen form, is explosive. So it's kind of important that you don't let that hang around. So what you do is you pick that up with something called a hydrogen acceptor. And we also get our energy, and our energy we're going to convert into ATP, okay? So we've got our hydrogen acceptor, which is going to become a reduced hydrogen acceptor. So that's just the name for saying it's got hydrogen attached. Okay. Right. So we're going to move on to the second stage. We're going to take our energy with us. We're going to take our hydrogens with us, and everything else has been used up. We don't have to worry about any of this. We're not going to use chlorophyll again. The water's been smashed up and the oxygen has left. Okay, so stage 2. So we're going to use the hydrogens, we've just said, and we're going to use the energy. Okay, so if you think about your original equation said water and carbon dioxide, and light goes to glucose and oxygen. Right, well, we've already used up our water, so it's no longer part of this. We've used our light, because that was part of the light bit, and our oxygen has also out of the equation. So it means in the second stage we can only be using carbon dioxide, and we're going to make glucose. Since this is called carbon fixation, that's maybe not surprising. We're going to fix this carbon here into a useful carbon for us. And glucose is C6H12O6. So you can see why we needed our hydrogens from the first stage. And we've obviously got oxygens in there. So we're going to get our glucose molecules, which we're then going to use for various things. Okay, it's the kind of basic chemical form of energy that is used on pretty much everything on Earth. So we're going to use a cycle. Now you're not going to need to know much about this cycle other than it's a cycle of reactions. At higher you need to know a bit more about it, but we're okay with a basic concept of this just now. So we're going to bring in our carbon. We're going to put it through a series of reactions. These are all going to be controlled by enzymes, and we're going to use up that energy that we created in the first stage. So once we've done that, this ADP and PI can just scoot back to the first stage, pick up some more energy, come back in. And we can do exactly the same with the hydrogen. So we bring in our hydrogen acceptors, we use them, and then our hydrogen acceptors that have now got no hydrogen on them zip off back to stage 1. Okay? And that's your lot. You have to flip round several times to make a glucose, and you're good to go.